Okay, we have a few more questions to deal with. The first one is this. How do mutations change phenotypes? So how do mutations change the way an organism looks or the way an organism behaves? Okay, those are the two biggies of phenotype, looks and behavior. So mutations can come in the following forms. You've addressed some of these already on your quizzes. Actually, if you did the quiz for today, you've addressed all of these. First one, missense mutations. So a missense mutation is one in which an amino acid is changed. The amino acid is changed. The codon changes from one codon to another, and this changes the amino acid. Now, I want you to know this. A missense mutation might have a very, very small impact on the phenotype. Or it might have a very large impact on the phenotype. Like in these examples here, sickle cell anemia, albinism, hemophilia, achondroplasia. These are all genetic illnesses. Some of these with huge pathological implications. And so these are missense mutations that have a huge impact on phenotype. Some missense mutations, they might still change the amino acid, but it might have a very small impact on phenotype. For instance, let's say the codon originally coded for an amino acid that had an acidic R group, that the R group acted as an acid. Okay? You got that in your mind? Okay, and now you have a missense mutation, and that codon is now a different codon and is translated into a different amino acid, but that amino acid still has an acidic R group. So it's a different amino acid. It's a missense mutation, but chemically the two are similar. That might end up having a small impact on phenotype, but it's still different. So it's not going to be chemically identical in the same way that two acids aren't chemically identical, but it might have a small impact on the phenotype. Okay? So what I want you to take from this is that missense mutations have variable impact. Okay? Sometimes the impact is very large, sometimes the impact is very small. Okay? And many of you picked up on this for your quiz on Monday that asked, how can you have a mutation to not impact phenotype? And some of you are like, well, if it happens in a section of the DNA that doesn't get translated into a protein product, it might have no impact. That's true, even though it could very well be it could very well change that DNA quite a bit. All right, nonsense mutations. So nonsense mutations, they change one of the codons into a stop codon. So basically, you get what's called an early abortive sequence. So it stops the translation before it should have. So remember, there are only 3 stop codons, right? There are 3 stop codons. Codons that do not have an anticodon on a tRNA, and instead their anticodon is on a release factor. There are only 3 of them. But if you mutate a codon into one of those 3 stop codons, now your translation stops before it should have. And the likelihood of being able to still get a functional protein product with a nonsense mutation is almost non-existent. Almost non-existent. Nonsense mutations are almost always devastating phenotypically. It's not variable like missense. Missense can have a variable impact. Nonsense mutations is almost always a big impact. Yeah, Chris.
What's a disease or something like that is because of the nonsense mutation? Do you have an example?
Yeah, that's a good question. I'll have to think on that. I mean, so I can tell you that several of the mutations that cause cystic fibrosis are nonsense mutations. There are about 100 different mutations that can cause cystic fibrosis if you get 2 mutant alleles. You only need 1 functional allele to make the proper ion channel. Okay? But if you've got 2 of them, that's cool. But if you have 1 of them, that's cool too. You're still going to make the proper ion channel. But if you have 2 mutant alleles, you're going to not make that ion channel, and you're going to have a lot of issues in controlling water balance. And so some of those mutations, and again, there are over 100 different mutations in that channel, and some of them are nonsense mutations, where you just don't make the protein product. All right, and then silent mutations. Silent mutations, it changes the codon, but it's the same amino acid. Okay? And so we know that there are 64 different codons. 61 of them code for an amino acid, 3 for a stop codon, right? So there are 61 different codons translated into 20 amino acids. So there's a lot of redundancy. And it's that way for a reason, so that amino acids that appear very, very commonly have a number of ways in which you can translate them. Hopefully you're starting to get a picture that there are a lot of ways that our genomes control against change, control against alteration, right? That DNA polymerase fixes over 99% of the mistakes that it makes, right? It makes one mistake in, what did we say, 1 in 100,000 bases that it makes a mistake, and it fixes, those are that persist, okay? And so it's impressive. And then besides that, you've got DNA repair mechanisms that will fix over 99% of the ones DNA polymerase doesn't catch. And then there's ways in which you could leave a mutation, not catch it, and it not have any impact on the organism. That's good stuff. You're like, man, these are some serious obstacles to overcome, to have mutation be your only mechanism of getting variation. Right? If all life roots back to a single ancestor, mutations are your only source of variation. But organisms are designed to make mutations very difficult to persist. And then on top of that, at the population level, natural selection is really good at eliminating mutations. Yeah, like this whole thing seems to be designed in such a way to maintain balance and preserve life, and to protect against change. And yet, change is your only way that you get variation, because it can't be there from the beginning. Unless, of course, you have a creator that created a lot of this variation and put things in place to protect it, to protect the creation, make sure it continued to function. All right, frameshift. So frameshift mutation, it shifts the reading frame. It's a deletion or an insertion of bases. And so if you add like an entire 3 bases, you basically just added another codon. But if you only add 1 base, it shifts every codon, right? Does that make sense? If you only add 1 base and you read the bases 3 bases at a time to figure out how to translate them, and you add 1, it shifts the entire reading frame. And not just for that gene, but potentially everything that comes downstream of it on the chromosome. And so if this frameshift mutation happens pretty far upstream on a chromosome, it can change the entire chromosome and every gene on it. Okay? And again, these are very, very rarely anything other than devastating, these frameshift mutations. Okay? These are almost always devastating. It's not like Missense where there's a variable response. These are almost always devastating. Yeah.
You mean like devastating as in like really like expressed on the phenotype, or like death?
Both. Frameshift mutations would almost always lead to an inability to develop properly. Yeah. Yeah, Levi.
So with these missense mutations, what is the this variable result? What does that variation look like? Is it very often functional with the product?
Yeah, so if you switch it from one amino acid to a chemically similar amino acid, you're probably going to only see a difference under certain environmental stress.
How often does that happen?
It depends on the particular situation. Yeah, that's a good question. And it depends on what the protein does. If the protein is an enzyme, it's and it also depends on where the mutation happened. So if the protein is an enzyme, which we'll talk a lot about on Friday, proteins that fold into enzymes have a very specific chemistry to attract whatever the reactant is for the reaction they're facilitating. Okay? They're chemically perfect to attract that. And so if your mutation switches an amino acid in the section of the enzyme that binds that reactant, even if it's similar chemically, it's probably going to be devastating because it's not exactly the same. It's not perfect. But let's say you had a mutation in like a channel, something that facilitated the diffusion of a polar molecule through the cell membrane. If you change an amino acid into another polar amino acid, it's probably going to have no impact on the function of that channel. Because it doesn't have to be perfect, it just has to be polar, so that it creates this polar core for polar materials to move through. Does that make sense? Yeah. Cool. Yeah. Davies.
Which mutation would Down syndrome be classified in?
Down syndrome is none of these. Down syndrome is a chromosomal mutation. These are all what we call point mutations, where you mutate a specific section on the sequence. Down syndrome is the result of a chromosomal mutation. It's an extra copy of chromosome 21. But we'll talk about those types of mutations in a little bit. David and then Allison.
So for silent mutation, is it like really devastating?
No, silent mutations, there's no impact whatsoever, at least for that gene, because it changed the codon, but it's still translated in the same amino acid. Yeah. Allison.
So are these mutations that are on the DNA or are they copying errors from the RNA? Or can they do
Yeah, well, both would result in the same thing, but we're talking more here about permanent changes to the code. So like a copying error that changed the DNA, so that every time you write the RNA, it's giving you a difference. But if you just wrote the RNA incorrectly, then it's just a one-time issue, and the next time you write it, it'll probably be written correctly. And when you transcribe a section of the DNA, you usually don't just make one pre-mRNA, you make hundreds or thousands of them. Yeah, that's a good question. All right, so here's the normal gene. There's the DNA getting transcribed into RNA and then getting translated into a polypeptide. Doesn't become a protein until when? Till it's folded correctly and it's transported to the correct place. Right? Okay. Here's a missense mutation. So what was it originally? Oh, the mutated used to be TNA, now it's C and G. So it gets written into RNA differently. Instead of being a U here, it's a C. Should have been a U, but instead it's a C. And so it changed the amino acid. The codon changed and it was translated into a different amino acid. And again, depending on the chemical similarity between this amino acid and the other one is going to determine a great deal about the impact of this mutation. So here's the missense mutation for sickle cell anemia. A single change. Instead of being GAG on the mRNA, it's now GUG. Instead of being translated into glutamate, it is translated into valine, and it changes the chemistry of that molecule. And the molecule no longer holds oxygen properly. On top of that, the molecule becomes very attracted to other molecules of hemoglobin, and they cluster up. And then they make red blood cells take on this really awkward sickle shape, which is why it's called sickle cell anemia. Instead of being donut shaped, they're shaped like a sickle, like something you would, the Reaper has. And then they clot, you form blood clots a lot. And so this is a missense mutation with an enormous implication on the phenotype. You're like, well, why does it even exist? Why doesn't natural selection get rid of it? You just told us, Dr. Engel, natural selection does a great job of getting rid of these mutations. Here's the problem. You only need one proper copy to be normal, or mostly normal. So if you have one mutant copy, you're not going to have sickle cell anemia. You're going to be a carrier for it, but you're not going to have sickle cell anemia. So there's no way really for natural selection to keep you from reproducing. On top of that, people that are heterozygous for it, that have one normal copy, so one of their chromosomes would be transcribed into this, the other one is transcribed into this, they are not as susceptible to developing malaria. And so in areas of the world where malaria is really common, you find sickle cell trait really common. So in South America and Africa and Southeast Asia, where malaria is really common, you find this is a really common mutation. There are a lot of carriers with what we would call sickle cell trait. They don't have the disease, but they do have sickle cell trait. And they're mostly, I say mostly normal. If you get to really low oxygen levels, they have a hard time. Did we talk about this already? told you about James Harrison. He was a linebacker for the Pittsburgh Steelers, and then he finished his career playing for the Patriots. But whenever they went to Denver, he couldn't play because he was a carrier for sickle cell anemia, and at that high elevation, he couldn't breathe. So he would never make the trip to Denver. But other than that, he was phenotypically normal. Yeah.
Can people with sickle cell anemia, can they still take blood from other people? Does it not change their like antigen or antigen?
Oh yeah, it doesn't change the antigen, so they can still take a blood transfusion, and so you can treat sickle cell anemia, and you usually treat it, I think, with like a marrow donation. So you give somebody like normal, healthy, what we call hematopoietic stem cells that are capable of becoming all the different types of blood cells, and so they can make normal red blood cells. Yeah. So you can treat it, and you could treat it with blood transfusions, but you've still got a clotting issue. Yeah, Mia.
If carriers don't exhibit it phenotypically, then how can they still have trouble at higher elevations if they're?
So they do have some, because that's what we call a dosage compensation issue. That in most conditions, only having one normal copy is good enough.
Oh, good. Okay.
But when you have a lot of environmental stress, only having one copy is no longer enough.
So they still have a little bit of sickle cells.
Sure, you can look at it that way, but they now at high elevations they don't form like sickle cells. The red blood cells don't form those sickle shapes. They just don't carry oxygen as well.
Okay.
Yeah, they just don't carry oxygen as well. But I don't think anybody would argue that James Harrison is like unable to metabolize properly. I mean, he was a professional football player for a number of years, so he figured it out, right, to make it work, even though he didn't carry oxygen as efficiently as he would if he didn't have sickle cell trait. All right, here's a nonsense mutation. Here's the original pair was CG, now it's AT. So this should have been UGC, but now it is UGA. And UGA is one of our 3 stop codons. Okay? And so instead of being translated into an amino acid, there's no tRNA with an anticodon for this, and instead it brings a release factor too early, okay? And it terminates the process too early. And so we don't even get to build the entire polypeptide. All right? And then here, a silent mutation. Supposed to be a T here, so it should have been GGT, but instead it should have been GGU. There's no T in RNA. Sorry. You knew that, though. You're like, Dr. Engel, it's not a T. It should have been GGU, but it says GGC, but that happens to be translated into the same amino acid. So there's no impact on this protein. Okay? That's why we call it a silent mutation. It's a change. The DNA has been permanently changed, but the protein product has not. Yeah, Chris?
How did they find a silent mutation? Like, how do you
Yeah, what's interesting is when you compare like you and I for the same gene. Like, so you and I have obviously the same genes because we have the same needs, but your version of the gene might be a little bit different than mine, but they still make the same protein product. And so you compare my genome with yours, you're like, wow, there's some differences in this gene. And then you go and explore it further and you're like, oh, it's because the differences still result in the same amino acid.
Okay.
So it's part of what makes you you, you know? So that you and I are not genetically identical. Yeah. Because that would be really sad for you. I'm really, yeah, anyways, sorry. I'm really happy for you, Chris, that you are genetically distinct from me. Yeah. All right, so frameshift mutations. Here we inserted an extra set, an extra set. Each strand on the DNA molecule got one extra base, and so it shifted the entire reading frame. This should have been GGC, right? Should have been GGC, but now we added an extra A in there, so it becomes GAG, and that C that should have been in this position is now in the first position in the next codon, and it shifts the entire reading frame down one base. And so every single amino acid has the potential to be different. And that's why this is almost impossible that this is going to be a minor impact on the phenotype. Yeah.
So things like these just stop everything from functioning, just like the pill itself?
Potentially. Yep, potentially. It depends on where they happen. If they happen towards the end of a chromosome and it only impacts one gene or a couple of genes or even no genes, then they can go unnoticed and kind of pass on to the next generation. So, all right. Yeah, Davies.
How do they test for these mutations?
That's a good question. You would need to sequence a particular region of the chromosome. If you have a suspicion that somebody has a type of mutation because they're exhibiting characteristics of a particular genetic illness, you sequence just a portion of the chromosome of the genome. And so we can talk about the mechanisms of how to do that, but we can target which sections we sequence. You don't have to sequence the entire thing. You can sequence just a piece of it. Yeah, that's a good question. All right, next one. How do transposable elements impact phenotypes? And so here we're going to take our lecture break. And what I'm going to do is I'm going to tell you a little bit about what a transposable element is, and then I'm going to let you talk with those around you and see if you can come up with some ideas of what some of the consequences of these might be. So transposable elements are mobile genetic elements. They're parts of the genome that are basically designed to get cut out and reinserted. Oftentimes to get cut out of one section and then reinserted somewhere else. Okay, so they're mobile portions of the genome. Okay, sections of a chromosome that can get cut out, reinserted somewhere else. Okay, and so what I want you to do, take about a minute or a minute and a half, talk with those around you, and I want you to come up with what this might mean as far as the function of the chromosomes is concerned. All right? Is that all right? Does it make sense? And I guess you could just read the rest of the slide to answer the question, but don't do that. Don't do that. Okay? All right? Yeah, so what I want you to do is just answer the question of what might the consequences of mobile genetic elements be? Parts of the genome that are designed to be cut out of one section and inserted somewhere else. Okay? 60 seconds starting now.
Taking a piece of code and putting it someplace else. So it's needed someplace
And then you miss the word, and then there is maybe imagine target site gets filled up.
Sure, like it needs a specific set of codon to make a polypeptide, but that specific set might be only found up here in this chromosome, so it'll take a copy of or cut. Yes,
Okay. So first off, just a little bit of information for you. We find these in basically all organisms. Basically all organisms on Earth have portions of their genome that are mobile, that can move from one place to another. What might some of the implications of that be? Yeah, Tara.
Implication, not consequence, but doesn't it help with genetic variability?
Oh, absolutely.
So even from the same organism, you can get different combinations.
Yeah, if order matters, the position of something is important, right? And order matters in the genome, right? It's not like, you've got 5 people, how many different, committees can you make of 3 people, right? It's like, no, order matters. Like, order matters in this situation, in the genome. And so if you change its position, it might change the way you read something. Absolutely. Any other thoughts? Micah?
Well, could it be that if there's a mutation in one place, you can replace that with one of these mobile?
Sure. Yeah, potentially. Yeah, Allison.
Is this similar to how bacteria incorporate plasmids?
So plasmids are rarely incorporated into the entire genome. They're usually left alone as their own separate entity. This is actually incorporating pieces of DNA into the genome. But it was already there, but it's capable of moving somewhere else. Capable of moving somewhere else. It's a good question. Yeah, Davies.
Well, since the genome or the genomic makeup of an organism kind of affects the phenotype, so I think when there is that cutting and pasting, sometimes there might be an error in one place.
Yeah.
So that might cause also like a mutation.
Yeah, it really depends where did it come from and where does it go, right? And where does that cut actually happen? It's great to bring up the cut and paste, which is the language that's going to be talked about a lot with these transposal elements. You basically have two types of transposition. You have cut and paste and you have copy and paste, okay? And you're like, okay, I understand what those mean. Cut and paste, I cut it from here and I paste it somewhere else. But where you do that cut has a huge impact on what that message is, right? So let's imagine for a moment you typed out a letter to somebody, okay? You typed out a letter to somebody, and then afterwards you're like, ooh, I really didn't want to say that, but I want to keep a copy of it in case I have to send a second letter, right? You're like, this is probably not the best way to freeze it. So you cut it out and you paste it somewhere else, you keep it, you store it away for, safekeeping later on. Where you cut is really, really important, right? Do you agree? Like, what you cut out is really important. And then as far as if you're pasting this in somewhere else, because it's not like we're taking something we're cutting out, we're like, ooh, I didn't really like the way that sounded. Let me cut that out of this letter. But we're doing that, but we're pasting it into another letter, right? And so where you put it in is also going to have a really big impact, right? Especially think about it if it's like a complete thought and you insert it right into the middle of another complete thought, now something really weird happens, right? And so where it reinserts has a big impact on what the phenotypic result is. If it reinserts in the middle of a gene, it destroyed that gene. If you cut out right in the middle of a gene, you destroyed that gene. And so now you're like, wow, this is really weird. If there's all this potential for wrong, why are there these mobile elements? Why are they? Why do they? And if they're found in virtually all organisms, it seems to be something that's part of the design. So what's the value of them? I'm glad you asked. Let's talk about some of this. So if the transposable element moves to a different position on the chromosome, let's say it left chromosome 1 and reinserts on chromosome 4. So it didn't leave chromosome 1 from your mom and then reinsert in chromosome 1 from your dad. Okay, it left chromosome 1 and reinserted on a different chromosome. We call that a transposition. It's moved. Its location in the genome has moved. And then there's two ways that this can happen. Cut and paste. It got cut, reinserted somewhere else. Or copy and paste. It gets duplicated and reinserted somewhere else. Gets duplicated and reinserted somewhere else. Yeah.
What does non-homologous mean?
Non-homologous means not the same chromosome. Because, in every cell in your body, not every cell, in almost every cell in your body, you have two entire copies of your genome, right? One copy that came from your dad, one copy that came from your mom, right? Y'all okay with that? Now, if you're male, it's a little bit different because the two sets are not exactly the same because male mammals have one X chromosome and one Y chromosome. But you basically have two complete sets of the genome. If something is removed from your maternal chromosome 1 and switches places with the paternal chromosome 1, that would be a homologous transfer. Okay? We're talking about the movement, say, from chromosome 1 to a completely different part of the genome, a completely different book. And you can think of those chromosomes as individual books. Chromosome 1 is its own book. Chromosome 2 is its own book. Chromosome 3 is its own book, so on and so forth until you get through all 23 sets. And then you just have two copies of each book. They're a little different. One copy came from your mom, one copy that came from your dad. But they're basically the same book, right? But if you pull something out of one book and insert it into a different book, that's obviously going to change the way things read because order matters. Just like when you're reading through a series of books, order matters, right? Let's think about this. Let's say, you take like the end of a story and you cut it out at the end and reinsert it in the beginning. That's like the most horrible story ever, right? Because you read the end and you're like, no, that's how I read stories anyways. I read the last chapter of a book and then I start over. No? Or maybe you don't read. Have you seen a book? You know what a book is? Okay. I just want to make sure we're communicating the same language. I'm sorry. I'm sorry if I offended you.
Question?
It was intentional, and that's what I'm sorry about. Yeah.
Is there a mutation where you have XXY?
Yes. Yeah, we'll talk about that. It's called Klinefelter syndrome. Yep. Yeah, we'll talk about that. You save that question for later. Okay, so here's cut and paste. We took something out of the chromosome here and we reinserted it somewhere else. And you're like, that's so cool. That is so cool. Why does it happen? I don't know yet, but it is cool. And here's copy and paste. So we didn't cut it out, we left it there and we just copied it somewhere else. You're like, now this is really cool. Okay. Any questions about that? So the last two questions we need to deal with is how do transposable elements impact bacterial chromosomes, bacterial genomes? How do transposable elements impact eukaryotic genomes? Okay. Yeah, Davies.
Well, I want to know what cuts them out.
A machine. There's a molecular machine that will cut them out and reinsert them somewhere else.
Does it have a name?
Yes. Yes, it does. But I can't think of the name of the machine right now. I'm sorry.
Oh, it's
I should. I should. I mean, here it just calls it the transposition mechanism, but I guess you can call it that. That's a very general term. The name of the gene is probably a combination of letters and numbers that really means nothing, but it's just how we categorize the gene. So how do bacterial genomes change from transposable elements? Well, bacterial cells have two types of transposable elements. First type, we call these insertion sequences. Insertion sequences. And these basically only have the gene for, oh, here it is right here, transposase. Yeah, the enzyme. So these insertion sequences have a single gene for the protein transposase that cuts these sections out and moves it somewhere else. Then you have transposons. And these have the insertion sequences and then have a lot of DNA in between them. Lot of DNA. And the interesting thing is here, we find this often in the portions of bacterial genomes that code for some product that gives them antibiotic resistance. And it looks like it's a way in which these bacteria are basically designed that they can cut the section of their chromosome out that gives them resistance to an antibiotic, reinsert it into a plasmid that then they can transfer to another bacterium to give another bacterium the antibiotic resistance. Isn't that cool? You're like, man, that's so kind of them. Like, they're rocking life because this whole colony of bacteria are being treated with an antibiotic. This one's doing fine because it's resistant, but it's not content with that. It wants to make other individuals around it resistant. So it cuts that section of its chromosome out, reinserts it in a plasmid, and then transfers that plasmid through conjugation to other bacteria. Yeah, Levi.
How does it know which?
It doesn't have to know because it's programmed inside of it. Their genome is programmed in such a way that it only happens in a certain area of the genome. It looks as though they've been designed to share certain features with other individuals. You're like, this is really weird. Why would that be? Unless, of course, you designed your creation to function in such a way where to move material from one individual to another did not require eating that other organism, right? So if what you believe about origins basically holds to there's no death prior to the fall, you need a way to connect ecosystems without feeding relationships, right? Because when we talk about ecosystems, what do you see as the model for how an ecosystem works? It's a food web, right? You have all these producers, and they're eaten by all these primary consumers, which are eaten by all these secondary consumers and all of these tertiary consumers. If that wasn't a reality to the original creation, you need a way to move material around and connect ecosystems without organisms eating other organisms? And what if you designed it in such a way that portions of genetic material can move from the organism from one place in its genome to another, or maybe from its genome to the genome of another organism? Now you're like, wow, this is actually kind of cool. You can make it to where as life spreads out after some creation event, or after, say, a global flood, some hypothetical global flood, right? That as life spreads out and you get dozens of different species ending up in the same place at the same time, and they all need a certain feature in order to survive there, that they actually have a mechanism by which they can transfer the code to make that machinery from one organism to another. You're like, wow, now it sounds like really good design. And a dominant idea right now is this is where viruses started. That viruses began as these transposable elements, and then they basically developed a way to live rogue. Although, quote, live, because you could argue that viruses aren't really living organisms, but to actually exist outside of the host. But it started as that host DNA. Do you remember I told you, I don't know, maybe a couple of weeks ago, that it looks like the part of our genome that codes for the machinery to help us fight viruses was inserted by a virus? I told you that a couple of weeks ago. But what if it was never originally a virus, it was always just a mobile portion of our genome that would move from one portion of your genome to another portion to ensure that it is always efficiently made into the machinery necessary to fight disease. And then somehow it gained the ability to actually exist outside of the host, and now you've got something really difficult to deal with. Anyways, just some interesting thoughts. So here's a representation of bacterial transposable elements. Here's a transposon where we've got these two insertion sequences, one at either end. In both of these, the gene for a transposase, the machine that cuts it out, reinserts it somewhere else, and then all of this central region with all of its genes. And so if your genome was designed in such a way that it cuts out like genes that, again, allow that bacterium to be resistant to antibiotics, now that bacterium can cut this out, put it into a plasmid, transfer that plasmid to another individual. And to answer your question, Levi, it doesn't have to think, it just does. Right? It's robotic. It's programmed to act in that way. It's like your computer, it's not thinking, it's programmed. You're like, no, my computer is thinking and it talks to me. We're friends. That's weird. And a little creepy. Sorry. All right, so how do eukaryotic transposable elements work? So eukaryotic cells, we've got transposons like bacterial cells do. And you're like, cool, we've got sections in eukaryotic genomes that code for transposase that will cut out sections and reinsert them somewhere else. You're like, that's cut and paste though, right? Cut it out and reinsert it. But what about copy and paste? Right? What about copy and paste? Well, eukaryotic cells also have what are called retrotransposons. Retrotransposons. And so basically what happens is that section of DNA gets written into RNA. And then what that RNA does is ultimately it gets translated into a protein that reads RNA and writes DNA from it. And so it reads the RNA, it writes a DNA equivalent of that, and then inserts it into the genome. And so it's like, it still stayed in its original position, but it got transcribed into an RNA that then got written back into DNA and inserted somewhere else in the genome. And you're like, now this is really cool because we can keep the original portion still functioning, but now we have a second version of it. And we're just really building up this potential to move sections of the genome other places. And you're like, again, what is the functional advantage of that? And it's like, I don't know, but it sure seems to be a great way to connect ecosystems if you have no feeding relationships. To have these mobile genetic elements that can move not only within organisms, but even from one organism to another. And you know what's interesting is somebody had this thought maybe 20 years ago. Somebody had this thought and wrote about it, and they were ridiculed because they're like, there's no evidence of horizontal gene transfer in eukaryotes. But you know what we found in the last 5 years? Overwhelming evidence for horizontal gene transfer in eukaryotes. And we're like, man, it really does look like eukaryotes can transfer pieces of their DNA to other eukaryotes. And it might go through a virus intermediate, but we don't know. And then you've got things like tardigrades. Have we talked about tardigrades before? Woolly bears, moss piglets, tiny little animals that are basically indestructible. They live around moss, and whenever the water dries up, they go dormant. But then when the water comes back, they come back out and they look like 8-legged pigs or 8-legged bears. They're really cute, but they're microscopic. Anyways, somebody's like, I bet these things could survive space. So they took some water with moss piglets in it, tardigrades, and they wiped it on the side of the space shuttle. They sent the space shuttle up to the space station and then back, and then they scraped that part of the space shuttle and added some water, and the moss piglets, they came back. They reanimated. They're basically indestructible. And then you look at their genome and you're like, holy smokes, there are genes from this organism in here. There are genes from this. They're just like DNA hogs. They just take DNA. It looks like greater than 50% of a tardigrade genome came from other organisms. They're just like accumulated. They're like hoarders. They just take whatever they can find and shove it inside of wherever they can fit it. It's like my brother-in-law. I should call him a tardigrade. Oh, he'd like that a lot, I think. It's very nice, because I'm a purger. Like, I like to just throw things away. It's very nice to have somebody close in your family that's a collector. That's what he calls himself. He doesn't call himself a hoarder. He calls himself a collector. But it's so wonderful. We have a great relationship. I'm like, Trey, I have this thing. Do you want it? He said, absolutely. I just drive it over to his house and never have to think about it again. It's wonderful. Noah.
You answered the question.
Oh, you're welcome. I didn't even mean to do that. So Barbara McClintock was the scientist that actually discovered these retrotransposons, and she discovered that it actually led to the different coloration in Indian blue corn. That these sections that they would copy and then paste other places, and it disrupted the ability to make various pigments. Sometimes they'd insert in a place that cut off like the yellow gene, and it'd make blue corn, or sometimes it'd insert it in both places to cut off the yellow and the blue, and it'd be white, and you get this nice variation in color. And you're like, what's the advantage of that for the corn? And it's like, well, it does taste good. It doesn't really taste it tastes a little bit different than normal corn, but it makes some really nice looking tortilla chips, right? Where you can get a basket of tortilla chips. Some of them are blue, some of them are yellow, some of them are white, some of them are even orange. But the beauty of this is now it allows this plant to attract different animals to move its seed around. Because some animals are attracted to this color, some animals are attracted to this color, some animals are attracted to this color. It's like, I'll just make them all, right? I'm not going to pick and choose. I don't want just the animals that like blue. I want them all. And they're going to come and they're going to eat that corn, and then they're going to poop the seeds out somewhere else and just spread that life cycle. You know, just keep it going. Keep it going. You ever wonder why corn doesn't really digest? It's because it's designed to be eaten by an animal and spread somewhere else. So that when an animal eats it and then they take it and the offspring of that plant doesn't compete with the parent for resources. Because like a raccoon came and ate that corn and then traveled somewhere else and pooped the little corn kernels out. You're like, oh, that is so wonderful. I never even wondered why I can't digest corn, but now I know, and I love it. I love everything about it, Dr. Engel. I'm going to tell everyone I know. Hey, do you know why corn doesn't digest? Do you know why corn doesn't digest? But you got to wait until you're like sitting at like a table with a lot of non-science people and just be like, hey, y'all, do you ever wonder why corn doesn't digest? I didn't either, but I know now, and you should know too. And so here's the retrotransposition. So this DNA gets written into an RNA, and then there's a reverse transcriptase that reads this RNA, writes it back into DNA. And then transposase takes that and plugs it in somewhere else in the genome. And you're like, now we've got all of these copies of these mobile places all over the genome. And then it's like, is the entire genome now mobile because this mobile element is spread all around? Maybe. And what a wonderful design. What a wonderful design. Like, I got something good, you want it, this is totally fine. I'll just cut it out of my genome and I'll give it to you. I'll give it to you, because I'm giving. But I'm gonna do the copy and paste so I keep it and give it to you. I'm not gonna do the cut and paste. Because if we do the cut and paste, I lose it, and it doesn't do me any good. All right? Questions? Thoughts? We usually don't get a lot of time to get to this point where like we finish our material and we've got a few minutes left of class, and then I'm like, hey, do y'all just have something you want to talk about, you know? I mean, I'm here, you know? You're like, yeah, yeah, Micah.
Question from the quiz. How does the target site split into two?
The target site split into two? Oh, that's a good question. So the target site is usually, it matches basically the other side of this. So at the ends of here, you've got something very much like the target site, and so it inserts it right in the middle of the target site.
Okay.
So the target site oftentimes is several dozen bases long, and in the middle of it, you get some nice homology with the ends of our transposable element, and so it works nicely to put it right in the middle. And it's a good question. Davies.
What happens when copying or cutting goes wrong with this?
Oh, yeah, I mean, you can destroy entire genes. Yeah. Yep, when copying or cutting goes wrong, yeah, you can destroy entire genes, which can be super duper devastating. There's also, this mechanism sometimes takes a version of chromosome 9 and puts it on the end of chromosome 22, and it takes a gene that helps the cell get through the cell cycle, and it puts it into a section of the genome that's expressed like crazy. And so it makes the cell start overexpressing a protein that helps it go through the cell cycle, and that's bad, because now the cell's going to go through the cell cycle even if there's an error. And it leads to cancerous growth. Yeah, Micah.
How does DNA replicate with this in place? Just the same.
Well, it doesn't really matter what the code is for during DNA replication, because no transcription's happening, right? While you're replicating the DNA, the only thing happening is DNA replication. Nothing else is happening.
But it replicates just the same.
Yeah, yep, absolutely. The DNA polymerase doesn't care what the order of bases is, because it's not it's reading them, but it's not paying attention. It's like I was gonna say it's like when you're sitting in the class and you might be writing what I'm saying, but you're not paying attention. But then that was like another mean thing to say to you, on top of like, do you know what a book is? You know. Maybe let's do this. It's like when I'm telling my kids something, and it's like they're nodding their head, but they're not really listening. It's kind of like that. That's what DNA polymerase is doing. It's just kind of acknowledging that there's words there and nodding its head and copying it, writing it down, but it doesn't pay attention to what the message is. It has no capability to see what the message is. No, you know what would be a better analogy? It's like you copying something written in a language you can't read, right? You can still copy the letters, but you can't actually read the language. You have no idea what the meaning is. You're just copying it. That's a better analogy and less offensive one. All right. Have a wonderful day. Enjoy the rest of your Wednesday.